All right, so happy Pi Day, everyone. It's March 14. Today will be a GenPals talk on getting started with SCENIC Plus analysis on HPC3. I think you guys all know me, but maybe so much on the internet does not. So I'm Tatyana. I'm a PhD candidate in Lawson Lab here at UCI, and I'm with the MCSB program fifth year. So this is the paper which published this method in Nature Methods last year. I had no part in it. I'm just presenting their package, and I'm going to use their tutorial as a guide, obviously not my original code. And today I will use my data, but it's unpublished, so it's going to be anonymized a little bit with just simplified labels. Some of you may already know that there's already a basic SCENIC package, not SCENIC Plus. That one was built several years ago for single-cell RNA-seq data, for inferring transcription factor-based regulons or gene regulatory networks using target gene and transcription factor co-expression, in addition to shared motifs in the target gene promoters. What's different in SCENIC Plus is that it's built for multiome, which combines RNA and attack data. And some parts of it work the same. It still uses target genes, transcription factor co-expression, shared motifs, but also looks at co-accessibility in the cis regulatory elements. So this provides a little bit higher quality inferences because we have one more thing to kind of cross-check as we're doing the analysis. It's talking about preparation of data for SCENIC Plus. Of course, it starts back in the lab. This is actually the most important part, garbage in, garbage out. So we have to start with good quality data starting in the lab. So there might be some optimizations that you have to do for tissue dissociation, nuclei isolations. In my data that we talk about today, it's from mouse model of a cancer. So I had to optimize some steps for that. Library construction is pretty straightforward using the 10x genomics protocol. Basically, start with nuclei, do some initial steps, then split the attack and gene expression into separate aliquots, sequence it separately, and then combine them bioinformatically later on. And you can do some downstream analysis before starting the SCENIC Plus. And I would actually recommend doing that because like SCENIC Plus is very computationally intensive. So the more you can narrow down your data that you're interested in really learning the networks for, the better off you probably be. For software installation, first request a interactive command line session the following way. Make a new Conda environment with this Python version. Will not work with any other version. And even though this package is run in Python, you really don't need to know this language very well. The package runs really well, at least this version does. So you won't need to go into the source code and modify anything. So you don't have to feel like you need to be an expert in Python to do this. You really don't. And then this is the installation step. Install it pretty fast. Ideally, we would have a containerized notebook for this, but we don't have that yet because I never asked for it. So for now, we're just doing it at command line. Command line has its limitations. Plotting is an issue. So if you do decide to try this at the command line, you have to make a couple adjustments. First, you have to install some sort of X11 forwarding software on your own computer, like Xcords for Mac, which I did, but I think MOBA Extern or something else for Windows. When you connect to the HPC, you have to make sure that you turn on the X11 forwarding at the connecting step through SSH. And then when you request the interactive session, you have to make sure to put this X11 flag there as well. So in two places, you have to specify X11 forwarding. Some of the plots still won't render this way. So you just have to save them and download them, look at them later. Unfortunately, not all plots rendered for me at least. Again, this is the tutorial that I used. So parts that I'll be talking about will come from this tutorial on 10x multiome, this step-by-step part, and also some velocity, unbranched GRN velocity. Right before starting, there's a couple more things you have to install. One is the blacklist regions bed file. This is for problematic regions of the genome. You can download it this way. You also will need an annotation table of the transcription factor DNA binding motifs. You will need 2 databases for scoring and ranking. They're really big, so give yourself some time to download these. Next is a list of genes which are transcription factors. And finally, this program to make bed files. Parts 1 through 4 are species specific, so you have to decide which genome build you're going to use for your model if it's not human, but this last one doesn't matter. It works for everything. A little bit about my data that I'll be showing today. So I have multiome and just regular RNA-seq for the same kinds of samples. So first, I did a lot of exploratory analysis on this single cell data and did some annotations. And then I performed label transfer onto multiome and then went on with these populations for the analysis. Since this is unpublished, but it's going on YouTube, I didn't want to put results on here. But I will say that these cell populations are involved in breast cancer. The most important one that we're interested in is A. There's a subpopulation of A, I'm going to call it A sub. There's population B that's kind of closer related, at least in our model. This cell type is also involved in cancer. And then population C is a little bit less related. It's still a similar type of cell, but it's different. So I hope you guys don't mind that I'm using A, B, and C. But really, if someone is not in the cancer field, these labels, if I even put real names on here, it wouldn't mean much to some people anyway. So I think A, B, and C is probably sufficient, I hope. Since I started my data, there's probably other ways to make data objects.
Sorry to interrupt, Tatyana.
Yeah.
I think you just froze for a second. Would you mind repeating the last 30 seconds or so? Just
Was it from here?
from the next slide.
From here?
Next slide. This one. Yes, perfect.
Oh, okay, sure. Yeah, so I was mentioning that I began working on the data in Seurat, which is run in R. So when I wanted to run SCENIC Plus, I had to convert it to something that's compatible with Python. So I made an data object from Seurat by splitting up the matrix metadata and the embeddings, and then building it into an data object. And you're welcome to try some wrappers that might do this for you, but I just did it this way. And I actually took this example code from one of Sam's tutorials, but it seems to work really well. So that's what I did. So here we're going to start talking about pre-processing for SCENIC Plus. It's a 3-step run. First 2 parts are pre-processing. The third is like when you actually run the package. So in this step, we're going to use systopic, the Python version, so it's Py systopic, for inferring enhancers. This is the first step is to annotate the attack peaks in the data using the RNA cluster labels. And if you happen to take the same route as I did by beginning in Seurat, then you might need to watch out that your barcodes match in the attack and RNA-seq. But if you just start from ScanPy, for example, then this is not something you need to worry about. Next step is to make suitable attack profiles, again, using those clusters that were selected before and that were used to annotate the attack peaks. And then identify the potential enhancers. There's a QC step, and then 2 steps for identifying the enhancers, which is first differential accessible regions, and then topics through topic modeling. So this is the QC step. There are 3 QC plots that are generated, and it's kind of iterative. The first time, I just ran it with their default cutoffs, and then I saw that it didn't quite work for mine, so I adjusted them. And the goal here is to select the double positive populations in the little plots over here. So overall, almost 8,000 of my attack fragments passed QC, and almost all the cells. I think I lost 5 cells out of 1,500. Otherwise, the vast majority of them passed the attack QC and the RNA part QC. In topic modeling, there's a simulation that you have to do to figure out the best number of topics to use. Those of you who attended Narcis's defense yesterday learned all about topics. They're kind of hard to explain, but in a way, this is kind of like determining a clustering resolution to move forward with. And this plot ideally would show that 4 of these different methods maximized at some point, but in my case that didn't happen. So I went with the number of topics that at least most of the model is maximized at. This step was intensive. I only put in 1,500 cells, but it took 45 minutes with 6 CPU cores, which was 36 gigabytes of memory. These were the results. On the left is a recalculated UMAP using attack data, whereas before I was showing you the RNA-seq UMAPs. And on the right here are the different topics that were identified for most of these clusters that are labeled before. Second step of the pre-processing is PySys target. So it's a sys target with Python. This part is for identifying cystromes of bound regions. So it looks at the different motifs that are significantly enriched in the differentially expressed regions. I'm not sure why they don't use footprinting for this to really make sure that something is, that the description factor is bound. But in any case, it's done this way. This is where all of those big databases and motif tables come to work. And this part was very intensive. I kept getting messages that the parallelized little, we call them raylets because the big package that does this is called Ray, like a sun ray, I guess. And whenever those workers get hung up and stop working, you get these messages saying a worker was killed or died. So at this point, you have to restart it. So here, I had to go up to 32 CPU because any less, it just crashed. So it's not that I was impatient that it took 30 minutes, it would crash. So I had to up the memory. Lastly, determine the biomart host that will have the highest overlap in genes with your data. And for data exploration later, you'll need to remember which host you used, so keep track of that. And then finally, put all those components together to run SCENIC Plus and infer gene regulatory networks. So this is the code here. And I just wanted to mention a couple of things here. Again, we're going to specify which clustering resolution, and this needs to be the same one that we used before. So this is like my label is ABC. For anything that's not human, you have to adjust, of course, these species-specific parts. This is the bed program from before. This location for parallelization dumps has to be specified in a very particular way, unfortunately, for run SCENIC Plus portion only. So for me, finally, it worked when I specified it this way. I'm so sharing it. And since this process takes a while to run, it tries to save whatever stopping point it got to so that if a crash happens, at least not everything was lost. So also this saving part is specified here. This was really intensive. When I tried 32 CPU cores, it crashed after 45 minutes. So I upped it to 64, which was an entire cluster node in the HPC, and that ran for 2.5 hours, but successfully. So at the end, this worked. And again, I wanted to really point out this temporary directory that was a big problem for me.
Wait, sorry, Tatiana, was
Yeah.
this really just for 1,500 cells?
Yes.
That's wild.
Yes.
Like, you said earlier you had like 5,000 that passed QC. Like, did you run
No,
them
no, it
in?
was 1,500 the past
It was
QC.
15, only 1,500. Okay.
Yeah.
Ooh. I'm just like, what if you had more cells? I'm just like, would you need more? Oh, yeah. Okay.
I
So
don't know.
that's
Maybe
just
adding more cells doesn't really like exponentially, increase your resources that you need. I don't know because I
yeah.
just did this one time, but yeah, just FYI,
Yeah.
it takes a lot.
Okay. All right.
How many features should pass?
Oh, Z asked, how many features do you have for attack seek?
Passing QC was like 7,000 something.
Okay, 7,000 something. Ooh, I think so. Yeah. All right. But yeah, sorry for interrupting.
Yeah, it's
Thanks.
okay.
Continue.
This is a couple of example plots from the results of running SCENIC Plus. So on the left is a UMAP. And what's very interesting here is that population C, which was not very closely related, is very distinct here, whereas the other populations that were involved kind of got to be one big blob together. So I probably should rerun this without population C to get better resolution of this one. Knowing our mouse model and the cancer that we're studying and stuff, this result actually makes sense because we have a problem with differentiation, and that's what causes cancer. So this was not a surprising result. It was actually a really, really cool result, I thought. But still, to try to understand the different subpopulations, I'll have to remove population C and rerun it with the rest. You can plot the network. So on the right is that. So we have the master regulator transcription factors here, and the regions that they're associated with, as well as the downstream genes. And to actually see those regions, you can turn on the region visibility by replacing the 0 with a different size. But then it becomes even harder to understand what's where. So maybe you don't have to do that part. But it was kind of weird that by default it wasn't there, since the regions, the attack regions, are such an important part of the analysis that in a plot they're not shown. So you can manually go turn it on to see it. There's one more kind of analysis you can do after running SCENIC Plus, and that's trajectory inference. So you can look at the Regulon velocity and the differentiation forces. And since I clearly need to rerun my analysis without this population C, my velocity doesn't look so great. But hopefully after I rerun it, we can see some velocity vectors going from the interesting population A sub to wherever it points to. I'm currently still working on some of the plots. For example, this integrated multi-ohm plot. You guys probably have seen variations of this if you work in multi-ohm data from Seurat and SCENIC and stuff. My issue is I'm trying to figure out a best way to select the regions and genes to look at, because in the results there are literally thousands of them. So it's kind of not clear how to select the best ones. In the tutorial, it's just shown as an example where they picked something, but it's not clear how to go about methodically picking the best one. So I'm still working on that. I wanted to comment on the novelty of the results you might get if you use this package. So I guess the question always comes up, do we need to use multi-ohm, or can we learn what, same thing with just regular RNA-seq, or, things like that. So in my case, the top regulons that came up actually were not found in the differential expressed genes. So if it wasn't for running SCENIC Plus, I actually wouldn't have picked up these transcription factors to focus on. So one of them, for example, the expression is kind of everywhere. I mean, it's not exactly uniform everywhere, but it wouldn't probably come up as differential expressed anywhere. Looking at the target genes, there is some differences between these, two clusters over here, and then differential expressed regions, also a little bit like that. So if I was just looking at expression, I would have, and I did, miss this particular transcription factor. I want to share a couple of bloopers with you guys from some issues that I, made, mistakes that I made from just being tired and copy and pasting stuff without paying attention. So there's a lot of different things you can specify as you're running the different parts of analysis. One of them, of course, is how many CPU you want to use. So I was just copy and pasting and kept their default of 5, whereas I was paying for 8 and, made the code run slower, and I wasted resources basically. And the next one, there's a part that I already mentioned that crashes a lot. And I didn't realize that when I restart, the software only looks at presence of certain files, but not their integrity. So as I was moving forward and just chugging through, those garbage files were messing up the analysis, and I ended up with an empty object. So I had to go back, clear out the garbage files that didn't get made correctly because the software crashed, and had to restart. And then basically the most important thing I did next time was request more memory, and then it worked. That is all I have as far as content. Wanted to acknowledge my lab, my PI, Devin Lawson, and almost co-PI at this point, Kai Kasemrock, and Sharmila, who made the libraries. They were paid for by the Cancer Center U54 grant. I had a lot of help from the HPC team, the Research Cyber Infrastructure Center, RCIC. We used some BioRender here. Also wanted to acknowledge the mice that we use in our lab. And also my husband, who helped with the first iteration with SCENIC Plus before the software was published, when they were still in bioRxiv. They were really difficult to work with, so I couldn't even install it. And he does software work, so he helped me install and go through the source code and even edit stuff that wasn't working. But now that's not the case anymore. The software works really well. So I encourage you guys, if you're interested, to give it a try. And also I have some funding sources I wanted to mention, F31 and G32. And if there are any questions, let me know. I'll be happy to discuss anything.
Yay, thank you, Tatyana, for the presentation and tutorial.